Welcome to Ultimate Sleep Doctor. Tonight, we'll be exploring a fascinating secret held within our very being. Consider the intricate dance of your DNA, the blueprint of you. But what if that blueprint wasn't the whole story? What if there were whispers, subtle influences shaping how your genes express themselves? Epigenetics reveals a hidden layer of control, chemical signals that turn genes on or off without changing the code itself. It's a delicate tapestry, woven by experiences, by our environment, by the love we share. Like ripples in a quiet pond, these changes affect us and those who come after. Let your worries soften and feel the gentle rhythm of your breath. Tonight, we unlock a layer of understanding about the profound connection between our experiences and our health. If you find these explorations soothing, please consider liking this video and subscribing to Ultimate Sleep Doctor. Together, we can soothe more wandering minds, one peaceful night at a time. Just breathe, let your thoughts drift. Let's begin. Long before we understood the intricate dance of DNA, before the double helix was even a glimmer in a scientist's eye, the question of how traits were passed down from one generation to the next had captured the imaginations of thinkers for centuries. It wasn't enough to simply observe that children often resembled their parents. People wanted to know why. This curiosity led to some truly fascinating and sometimes controversial ideas. One of the earliest champions of this exploration was Jean-Baptiste Lamarck, a French naturalist who lived in the late 18th and early 19th centuries. Lamarck proposed a revolutionary concept: acquired characteristics. He believed that organisms could actually change during their lifetime in response to their environment, and these changes could then be passed on to their offspring. For example, he suggested that giraffes had evolved long necks because their ancestors stretched their necks to reach higher leaves, and this stretching was inherited by their descendants. It was a remarkably intuitive notion, and for a time, it seemed to fit observations of adapting organisms. However, Lamarck's theory faced resistance, largely because it conflicted with the prevailing view of inheritance as a simple, direct transfer of physical traits. The scientific community was largely stuck in a world where inheritance was seen as a perfect copy, like a blueprint faithfully replicated from parent to child. But the seeds of a different understanding were being sown, largely through painstaking experimental work. A pivotal moment came in the 1940s with the research of Philip A. McCulloch and Ernest H. McNeil. Working with chicken embryos, they discovered that the amount of a chemical substance added to the developing chick could actually influence the development of its thyroid gland. Specifically, more of the substance led to a larger, more active thyroid, responsible for producing hormones that regulate growth and metabolism. This was startling. It wasn't a change to the genes themselves, but rather a change in how the genes were expressed, affecting the overall development of the organism. This suggested that the environment could directly influence the fate of a developing embryo, a radical departure from the purely genetic inheritance model. As the 20th century progressed, the concept of epigenetics, meaning above genetics, began to take shape, largely thanks to the work of one brilliant scientist, Conrad Waddington. Waddington, a zoologist and developmental biologist, was struggling to understand how genes, which he considered the fundamental building blocks of development, actually work together during the complex process of embryogenesis, the development of an organism from a single cell. He sought a way to visualize this intricate interplay. In 1968, he published a seminal paper introducing his epigenetic landscape model. Imagine a hilly terrain, Waddington suggested. The genes were like valleys within this landscape, and the state of each gene, whether it was on or off, was like a position on that terrain. During development, the embryo navigated this landscape, moving towards the valleys that represented the desired developmental pathways. Small environmental influences, such as changes in temperature or exposure to certain chemicals, could push the embryo's trajectory up or down the hills, ultimately determining which genes were activated and which remained silent. Waddington's landscape wasn't just a pretty picture. It provided a powerful framework for understanding how genes could be regulated by the environment. It suggested that development wasn't simply a matter of genes turning on and off in a predetermined sequence, but rather a dynamic process of navigating a complex terrain of possibilities. The landscape was especially helpful in explaining how seemingly similar embryos could develop into different individuals, depending on their unique environmental experiences. Initially, the epigenetic landscape model met with skepticism. Many scientists were hesitant to accept a concept that seemed to challenge the central role of DNA in inheritance. The idea that environmental factors could have such a profound influence was difficult to swallow, particularly given the established dominance of the central dogma of molecular biology, which emphasized the flow of information from DNA to RNA to protein. However, Waddington's model proved incredibly influential, and over time, as more evidence accumulated supporting the idea that environmental factors could alter gene expression, his concept gained increasing acceptance. Experiments demonstrated that changes in diet, stress levels, and exposure to toxins could indeed lead to heritable changes in traits, not through alterations in the DNA sequence itself, but through epigenetic modifications, chemical tags that attach to DNA or histones, proteins that package DNA, affecting gene access and activity. The slow, incremental accumulation of evidence, combined with the elegant simplicity of Waddington's landscape, transformed the field, laying the foundation for the explosive growth of epigenetics as a vital area of scientific inquiry. Let's delve now into one of the most extensively studied epigenetic marks, DNA methylation. It's a fascinating process, a tiny chemical change to our DNA that can have a huge impact on how our genes behave. At its core, DNA methylation involves the addition of a small chemical group called a methyl group, often derived from carbon, to a DNA base, most commonly cytosine. Cytosine is one of the 4 building blocks of DNA, and when a methyl group attaches to it, it creates what's known as a methylated DNA sequence. This seemingly simple addition dramatically alters the way genes are read and expressed. The machinery responsible for this process is remarkably complex and beautifully orchestrated. Several enzymes, collectively known as DNA methyltransferases or DNMTs, carry out the methylation. In mammals, there are 3 main DNMTs: DNMT1, DNMT3A, and DNMT3B DNMT. One acts primarily as a maintenance methyltransferase, faithfully copying methylation patterns from one generation of cells to the next during DNA replication. It's like a meticulous scribe, ensuring the methylation marks are preserved. DNMT3A and DNMT3B, on the other hand, are involved in establishing de novo methylation patterns, essentially creating new methylation marks during development. Their activity is heavily influenced by factors like signaling pathways and interactions with other proteins. Researchers are still unraveling the exact mechanisms that control DNMT activity and precisely when and where these enzymes are active. It's important to note that not all cytosines are equally susceptible to methylation. The location of the cytosine on the DNA strand dictates the type of methylation that occurs. CG methylation is the most common type, found predominantly in regions of the genome rich in cytosine and guanidine, hence the CG designation. However, other types exist, including CHG methylation, associated with guanine, and, crucially, CHH methylation, associated with hypoxanthine. The discovery of CHH methylation was a groundbreaking moment. Initially, scientists believed methylation only occurred in CG-rich regions, but the ability of DNMT3B to methylate CHH sites opened up a whole new realm of epigenetic regulation, particularly in regions of the genome previously thought to be largely unmethylated. The consequences of DNA methylation are profound. A primary role is to silence genes. When a DNA segment is heavily methylated, it physically blocks the binding of proteins needed to transcribe or read the gene's instructions. It's as if a tiny padlock locks the gene in an inactive state. This silencing is vital in development, ensuring that certain genes are only expressed at specific times and in specific tissues. Genomic imprinting provides a striking example of methylation's importance. This phenomenon involves the monoallelic expression of genes, meaning only one copy of a gene is active, while the other is silenced through methylation. The IGF2 gene is a classic example. Its maternal allele is silenced by methylation, allowing the paternal allele to be expressed, which is crucial for growth and development. Researchers initially discovered this intricate regulation in Drosophila fruit flies, and subsequent studies have confirmed imprinting in mammals like mice. Another key role is seen in X chromosome inactivation. In female mammals, each cell contains 2 X chromosomes, but only one is active. The inactivation of the other X chromosome is mediated by extensive DNA methylation, primarily occurring at the ZIST gene, which then spreads a repressive chromatin environment across the entire X chromosome. The link between DNA methylation and cancer is particularly compelling. Aberrant methylation patterns, often involving hypermethylation of tumor suppressor genes, are frequently observed in various cancers. For instance, studies have shown that silencing of genes involved in cell cycle control and DNA repair through increased methylation is a common mechanism driving tumor progression. Landmark research by Harley and colleagues in the early 2000s demonstrated that cancer cells exhibited widespread DNA methylation changes, highlighting it as a key factor in tumorigenesis. Furthermore, epigenetic drugs that target DNMTs are now used clinically to treat certain types of leukemia and lymphoma, illustrating the tangible impact of manipulating methylation patterns. Ongoing research continues to explore the intricate interplay between DNA methylation, cancer development, and potential therapeutic interventions. Deep within the cell, nestled amongst the tangled threads of DNA, lies a remarkable secret, a dynamic, ever-changing layer of regulation called epigenetics. While DNA methylation, as we've explored, provides a foundational on or off switch for genes, it's only one piece of a much larger, more intricate puzzle. The story of how genes are truly controlled unfolds through a series of chemical modifications to the proteins that package and protect DNA. Histones. These modifications aren't about changing the DNA itself, but about altering the very way the DNA is organized, influencing whether a gene can be accessed and ultimately, whether it will be read and used by the cell. Histones are proteins, and they're incredibly important because they form a spool around which DNA wraps, creating a structure called chromatin. Think of it like winding thread around a spool. The more tightly the thread is wound, the more compact the structure, and the harder it is to access. Histone modifications act like tiny adjustments to this winding, either loosening or tightening the grip of the DNA on the histone. One of the most common modifications is acetylation. Histone acetyltransferases, or HATs, are the enzymes responsible for adding acetyl groups to histones. This is like adding a little lubricant to the spool. It relaxes the interaction between the DNA and the histone, making the DNA more accessible and promoting gene expression. Conversely, histone methyltransferases, or HMTs, attach methyl groups. The effects of methylation are far more varied and can either activate or repress genes, depending on which amino acid on the histone the methyl group attaches to. Some methylation patterns encourage gene expression, while others act as a signal for silencing. Phosphorylation and ubiquitination are other key modifications. Phosphorylation involves adding a phosphate group, often triggered by signaling pathways and impacting gene regulation in response to external stimuli. Ubiquitination, meanwhile, attaches ubiquitin, a small protein, which can tag histones for removal or alter their interactions with other proteins. Sumylation, another modification, is a more recently discovered process that also affects chromatin structure and gene expression, often involved in regulating the recruitment of specific proteins to DNA. These modifications don't happen in isolation. The cell has evolved a sophisticated system of reader proteins that recognize and bind to these histone modifications. These reader proteins are like specialized detectives, scanning the chromatin landscape for specific signals. For example, proteins called bromidamine recognize acetylated histones, while others called PhD fingers bind to methylated histones. These interactions then recruit other proteins involved in transcription, effectively switching genes on or off. The study of histone modifications has provided stunning insights across a range of organisms. In yeast, researchers have identified specific histone modifications that play crucial roles in regulating metabolic pathways. In C. elegans, scientists have shown how changes in histone modifications can dramatically alter development and behavior. And in mammals, the complexity of histone modifications is truly astounding, implicated in everything from brain development to immune responses. For instance, modifications to histones within the hippocampus are linked to learning and memory, demonstrating the profound impact of this layer of regulation. Researchers continue to uncover novel modifications and reader proteins, constantly refining our understanding of this intricate system. The dynamic nature of histone modifications, constantly being added, removed, and interpreted, makes them a cornerstone of epigenetic control. providing a flexible and responsive system for the cell to adapt to its environment. The story of epigenetics isn't just about DNA itself, but about the intricate ways DNA is managed, influenced, and read. While DNA methylation and histone modifications are crucial players, a surprising group of molecules, non-coding RNAs, are emerging as key orchestrators, fine-tuning the epigenetic landscape with remarkable precision. These RNAs, unlike messenger RNA, mRNA, that carries instructions for building proteins, don't directly encode proteins themselves. Instead, they perform a diverse range of regulatory functions, acting as tiny conductors in the symphony of gene expression. Among the most well-studied are microRNAs or miRNAs. These short RNA sequences, typically around 2123 nucleotides long, patrol the cell's nucleus, searching for specific messenger RNAs, the transcripts produced from DNA. When a miRNA finds its target mRNA, it binds to it, often leading to the mRNA's degradation or blocking its translation into a protein. This is a remarkably efficient way to silence genes. For example, researchers have identified miRNAs that regulate development by controlling the expression of genes involved in cell differentiation, demonstrating how these small RNAs can dramatically impact the trajectory of a developing organism. Studies in plants and animals alike have revealed the critical role miRNAs play in responses to environmental stresses like heat or drought, highlighting their adaptability. Then there are long non-coding RNAs, or LNC RNAs. These are much larger than miRNAs, often exceeding several thousand nucleotides in length. LNC RNA are incredibly diverse, and their functions are still being actively investigated, but they've been shown to participate in numerous epigenetic processes. Some LNC RNAs act as scaffolds, bringing together different proteins involved in chromatin modification. Others function as decoys, binding to proteins and preventing them from interacting with DNA. For instance, a specific LNC RNA might bind to a histone-modifying enzyme, effectively controlling where and how that enzyme acts on the chromatin, thus altering gene expression patterns. Furthermore, LNC RNAs can be physically associated with specific DNA regions, guiding methylation machinery to precisely target particular genes. Their involvement in regulating imprinted genes, where gene expression depends on its parental origin, is particularly fascinating, showcasing their role in complex inheritance. Another fascinating group of non-coding RNAs are piRNAs, which stand for Pui-interacting RNAs. These RNAs are primarily found in germline cells, the cells that give rise to sperm and eggs, and their main role is to maintain genome stability. Many DNA sequences, called transposable elements, are prone to jumping around the genome, causing mutations and instability. piRNAs are critical in silencing these elements, preventing them from disrupting the genetic code. Pui proteins, which bind to piRNAs, recognize these transposable elements and trigger a cascade of events that lead to the silencing of these mobile genetic elements. This process is particularly important during early development when the genome is vulnerable to disruption. Scientists have discovered that piRNAs are conserved across diverse organisms, from plants to humans, underlining their fundamental role in ensuring genomic integrity. The combined actions of miRNAs, LNC RNAs, and piRNAs demonstrate that epigenetics is not simply a matter of DNA modifications. It's a complex network of interactions, with these non-coding RNAs at the heart of it all. They provide a level of precision and responsiveness that was previously unimaginable, expanding our understanding of how genes are regulated and how the environment can shape our biological destiny. Research into these RNAs continues to uncover new mechanisms and pathways, promising even greater insights into the dynamic and adaptable nature of the epigenome. The intricate dance between our genes and the environment has a surprising partner in crime: epigenetics. And when it comes to cancer, this partnership takes on a particularly complex and often alarming role. It's not simply that cancer cells have different genes, they're fundamentally operating with a different set of instructions, largely due to alterations in how our DNA is packaged and controlled. One of the earliest and most consistently observed changes in cancer involves DNA methylation. Normally, our DNA is decorated with methyl groups, primarily on cytosine bases, a process that helps silence genes and maintain proper cellular function. However, in many cancers, we see a phenomenon called hypermethylation, an excessive addition of these methyl groups. This frequently targets tumor suppressor genes, genes that normally prevent uncontrolled cell growth, effectively switching them off. Simultaneously, cancer cells often exhibit hypomethylation, a decrease in methylation levels, particularly on oncogenes, genes that promote cell division. This deregulation allows oncogenes to become abnormally active, fueling the cancerous transformation. The balance is completely disrupted, creating a cellular environment that encourages relentless growth and division. But DNA methylation isn't the whole story. Histone modifications, the dynamic modifications to the proteins around which DNA is wrapped, play a crucial supporting role. Histones are like spools, and attaching various chemical tags, such as acetylation or methylation, to them dramatically changes how tightly or loosely DNA is packed. Acetylation, for instance, generally relaxes the DNA structure, making genes more accessible for reading and expressing. Conversely, certain types of histone methylation can tighten the DNA, restricting gene access. In the context of cancer, we see a sustained increase in histone modifications that favor a more open chromatin structure, allowing for the uncontrolled expression of oncogenes and the evasion of normal cellular controls. Interestingly, enzymes called histone acetyltransferases, HATs, are often overly active in cancer cells, relentlessly adding acetyl groups, driving this open chromatin state. The influence extends beyond simple gene activation. Histone modifications are intimately linked to the process of metastasis, the spread of cancer cells to distant sites. Specific epigenetic signatures have been identified in cells undergoing metastasis, indicating that changes in histone modifications contribute to the ability of cancer cells to detach, invade surrounding tissues, and ultimately colonize new areas. The fascinating aspect is that epigenetic changes aren't just passively occurring consequences of cancer. They can also be used as powerful diagnostic and prognostic tools. Researchers are now identifying specific patterns of DNA methylation and histone modifications in blood, urine, or tumor tissue that correlate with the stage and progression of the disease. These biomarkers offer the potential for earlier detection and personalized treatment strategies. Finally, the growing understanding of epigenetics has led to the development of epigenetic therapies. Drugs like DNA methyltransferase inhibitors, such as azacitidine and decitabine, work by blocking the activity of DNMTs, the enzymes that add methyl groups to DNA. Similarly, histone deacetylase inhibitors, like vorinostat, block the activity of HATs, reducing histone acetylation and promoting a more tightly packed chromatin structure. While these therapies have shown promise in treating certain cancers, particularly acute myeloid leukemia, their effectiveness is often limited, and resistance can develop. Ongoing research is focused on developing more targeted epigenetic therapies and combining them with conventional treatments to maximize their impact. The future of cancer treatment may well hinge on harnessing the power of this intricate epigenetic landscape. The story of epigenetics is full of surprises, and today we're diving into a particularly fascinating area: how experiences can shape not just you, but also your descendants. We've learned how DNA itself, the blueprint of life, can be modified, and how these changes, called epigenetic marks, control which genes are turned on or off. But what if those changes didn't just disappear after a few generations? What if they could be passed down, influencing the lives of those who never experienced the original event? This is the realm of transgenerational epigenetic inheritance, a concept that's challenging our understanding of heredity itself. The fundamental idea is this: despite the unchanging DNA sequence, the instructions for gene expression can be altered and then transmitted across generations without a corresponding change in the genetic code. One of the most compelling lines of evidence comes from studies using C. elegans, a tiny, transparent roundworm. Researchers exposed worms to starvation, a stressful event, and then allowed the offspring to develop. Remarkably, the grandchildren of these starved worms exhibited similar stress-related behaviors, suggesting that the epigenetic marks associated with starvation had been passed down through the germline, the cells involved in producing sperm and eggs. Similar experiments have been repeated with mice. For instance, exposing pregnant mice to a high-fat diet resulted in their offspring, even when fed a normal diet, exhibiting increased susceptibility to obesity and metabolic problems. The key seems to be alterations in DNA methylation patterns, the addition or removal of chemical tags on the DNA within the sperm and eggs. These methylation patterns essentially remember the dietary experience, influencing the development of the next generation. Histone modifications play a part here too. Changes in how DNA is packaged within the cell, mediated by molecules like acetyltransferases and methyltransferases, can also contribute to this inheritance. However, demonstrating transgenerational epigenetic inheritance definitively is incredibly difficult, particularly in mammals. The complexity of mammalian development, with its extensive reprogramming of genes during embryonic development, makes it challenging to track epigenetic marks through the germline. It's like trying to follow a whisper across a crowded room. Scientists are focusing on the maternal environment, the conditions experienced by the mother during pregnancy, as a crucial factor in establishing epigenetic marks that are then passed on. Factors like nutrition, stress, and exposure to toxins can all leave an imprint on the developing embryo, ultimately shaping the offspring's gene expression. Researchers are exploring how the microbiome, the community of microorganisms living in the gut, might also play a role, influencing epigenetic modifications through various signaling pathways. It's a complex web of interactions, and much remains unknown. While the evidence is mounting, it's crucial to note that many observed effects are subtle and may not be directly attributable to epigenetic inheritance in every case. Further research is needed to unravel the precise mechanisms and the extent to which this phenomenon contributes to our overall health and susceptibility to disease. The potential implications for understanding complex diseases like diabetes, cardiovascular disease, and even mental health are immense. The story of transgenerational inheritance is far from complete, representing one of the most actively researched areas within epigenetics. It promises to reshape our understanding of heredity and, perhaps, even our perception of what it means to be human, a being whose past can subtly and powerfully influence its future. The universe of life is built on a blueprint, a magnificent sequence of DNA. But that blueprint alone doesn't tell the whole story. It's how that blueprint is read, interpreted, and ultimately expressed that creates the incredible diversity of all living things, and it's where epigenetics comes in. During development, a single fertilized egg divides and specializes, transforming into a complex organism with hundreds of different cell types. A heart cell, a brain cell, a skin cell, each with a unique job to do. Epigenetic mechanisms are the silent architects of this astonishing process, orchestrating the intricate dance of gene expression that defines cell fate. These mechanisms don't alter the DNA sequence itself, but they do change how accessible genes are to the machinery that reads and translates them. Think of it like adjusting the volume knob on a radio. The music genes is still there, but its intensity and clarity are controlled by the setting epigenetic marks. Key players here are DNA methylation, where chemical tags attach to DNA, often silencing genes, and histone modifications, which alter the way DNA is packaged, making it easier or harder for genes to be transcribed. These modifications aren't fixed. They can be influenced by signals, including those from the environment. Let's look at Drosophila, the fruit fly, as a particularly clear example. Consider wing development. Initially, the cells have the potential to become either wing or antenna, but through a series of epigenetic changes, specifically modifications to histones, certain genes are switched on, guiding those cells down the path of becoming wings. These marks are established and maintained, ensuring that the wing cells remain committed to their function. Environmental factors like temperature can even influence this process, subtly shifting the expression patterns and affecting wing size or shape. Zebrafish provide another compelling case. These fish have remarkable pigmentation patterns, controlled, in part, by epigenetic mechanisms. Different genes are activated to produce varying shades of color in different parts of their bodies, creating beautiful and complex designs. Researchers have identified specific microRNAs that target genes involved in pigment production, further refining these patterns. The beauty of this system lies in its responsiveness. The developing zebrafish can adapt its coloration in response to its surroundings, a phenomenon known as phenotypic plasticity. Furthermore, development isn't just a linear process. Epigenetic oscillators and feedback loops play a critical role in timing and regulating these changes. These rhythms, like biological clocks, ensure that events occur in the correct sequence, allowing for the precise timing of cell divisions and differentiation. For example, a gene might be initially silenced by methylation, but as it's needed for a particular developmental stage, histone modifications remove the silencing marks, allowing the gene to be expressed again. This dynamic interplay, controlled by feedback loops, allows for a sophisticated level of control over developmental processes. The study of developmental epigenetics isn't just about understanding how organisms develop. It's uncovering fundamental principles about how genes are regulated and how this regulation can be influenced by both internal and external factors. It's a field brimming with possibilities, offering insights into everything from birth defects to the potential for regenerative medicine. The field of epigenetics is rapidly evolving, driven by increasingly sophisticated technologies that are allowing us to peer deeper than ever before into the intricate workings of our cells. For years, scientists studied the genome, the complete set of DNA, as the primary determinant of who we are and what we can become. Now, we're realizing that the environment and how it affects our DNA plays a crucial role, leaving behind chemical marks that alter gene expression without changing the DNA sequence itself. This is where the future of epigenetics truly begins to shine. One of the biggest breakthroughs has been in sequencing technologies. Previously, mapping the precise location of DNA methylation, a key epigenetic mark, was a laborious process. Now, techniques like bisulfite sequencing allow us to identify exactly which regions of the genome are methylated with remarkable accuracy. Similarly, ChIP-seq, chromatin immunoprecipitation sequencing, enables us to pinpoint where specific proteins, like those involved in histone modification, are attached to DNA. These tools give us an incredibly detailed snapshot of the epigenetic landscape within a cell. But the most exciting developments are coming from the use of CRISPR-based epigenetic editing tools. Think of CRISPR as a precise molecular scissor, but instead of cutting DNA, it can be programmed to modify epigenetic marks. A particularly promising approach uses a modified version of the CRISPR protein, called dCas9, which acts like a GPS, guiding an enzyme to a specific location in the genome. Once there, the enzyme can then add or remove epigenetic marks, essentially rewriting the cell's instructions without altering the DNA code itself. Scientists are exploring ways to use this technology to silence genes that contribute to cancer or to reactivate genes that have been silenced by epigenetic changes. Beyond these targeted modifications, researchers are also delving into the complexities of the cell at a more granular level, with single-cell epigenetic analysis. Traditional methods of studying epigenetic changes often rely on averaging data from many cells, masking important differences between individual cells within a population. Single-cell techniques allow us to examine the epigenetic profile of each cell independently, revealing fascinating heterogeneity within tissues and organs. This is particularly important in diseases like cancer, where different cells within a tumor can have dramatically different epigenetic landscapes, driving variations in drug response and tumor behavior. The potential applications of this technology are vast, extending far beyond simply understanding disease. Researchers are exploring how epigenetic changes might be harnessed to combat aging, improve crop yields, and even enhance learning and memory. Understanding the epigenetic basis of developmental processes is also opening up new avenues for regenerative medicine. Perhaps one day we could reprogram cells to repair damaged tissues by resetting their epigenetic state. Of course, with such powerful tools comes a need for careful consideration. Epigenetic editing raises ethical questions about the potential for unintended consequences and the possibility of using this technology for enhancement rather than treatment. The prospect of altering the epigenetic landscape across generations, as explored in Part 6, further underscores the importance of responsible research and careful oversight. Ongoing discussions about guidelines and regulations are vital to ensuring that the future of epigenetics is one that benefits humanity while minimizing potential risks. The ongoing evolution of these technologies paints a truly remarkable picture for the future of this field, promising deeper insights into the very fabric of life. If you're still awake and enjoyed this exploration, please consider liking this video and subscribing to Ultimate Sleep Doctor. Together, we can soothe more wandering minds, one peaceful night at a time. The intricate dance of epigenetics, now gently understood, has unfolded within your thoughts. Let your mind drift, calm and unhurried, allowing your awareness to settle, inviting sleep to find you. Good night.